Welcome back. Today we are starting with chapter 2. I want to talk a little bit about some study tips for our class. So one of the benefits about being online and being asynchronous means that you can re-listen to lectures, right? So you can listen to me again and again and again if you want to. And some people have found that listening while walking or exercising, while you're in traffic, maybe in the car, during cleaning or chores can really help get this material into your brain and have another chance to think about it. Some students have also found that watching videos at a faster speed or a slower speed can help. So if you're finding, find something that works for you, right? That'll help you listen and engage with the material and make sure that you are getting the most you can out of it. Okay. And today we're talking all about chemistry, chemical composition of the body. That's what chapter 2 is all about. So again, we're starting with that kind of molecule place and building up to this physiology, organismal level biology. Okay. And we're talking about lots of different compounds. I'm actually going to split this video into two. So you'll have two chemistry videos to watch because we talk about all these different kinds of things we find in our bodies, right? Water, acids, bases, pH, buffers, carbohydrates, fatty acids, proteins, DNA and RNA, and ATP. Maybe some caffeine if you're anything like me. So lots of things to talk about in this chapter. So it'll be divided into two. Okay. So here's the first one. This chapter, though there's a lot in it, is review, right? You've already had a chemistry class as a prereq for this class. So it might have been a while ago, but try to recall some of the chemistry you learned in your chemistry class and realize that for biology, we're working with organic molecules. So we have a lot less things to worry about than chemistry as a whole, right? We're thinking about the chemistry that's important to biology. So go back and remind yourself about atoms, chemical bonds, molecules, and functional groups of organic molecules. So when I mention a phosphate in the class, talking about ATP, you should know what that is and what that looks like chemically. Okay. So we'll talk about the most important chemistry for this class today. Okay. But go back and review if you're finding that chemistry is something that you haven't looked at in a while. Okay. All right. So this is not something to memorize, but just to be aware of how much water is really in our bodies, is that the human body as a whole is the majority water, right? But some tissues, looking at individually, have more water than others, okay? So your brain is a lot of water, your blood is mostly water, right? And then looking at, it's kind of interesting, look at bones, only 22% water. Right? So some have a lot more dry mass than others, some have a lot more wet mass than others. The other thing that's interesting is that water content in your body changes throughout your life. Let's pull this down a little bit. So when you're a child, especially a young child, your body has a lot more water in terms of percent composition than it does as you get older. And so this just decreases throughout your life until you're older and your body's only about 50% water. Okay? And then we'll talk about this too, but we'll talk about intracellular fluid, so inside cells, outside celled extracellular fluid, and that can be divided up into interstitial fluid, blood plasma, and cerebrospinal fluid. But you'll notice is that a lot of it's intracellular and a lot of it's interstitial, so just between cells. Okay. Okay. So water is actually an inorganic molecule. It's polar, and it's required for all life, as far as we know. Okay. So some of the major components and things to think about, be reminded about with water is that here's a water molecule, right? H2O. So one oxygen and two hydrogens. Cohesion is really important in water. And what this means for cohesion of water is that it can create surface tension, right? So here's an example of a water strider, an insect that uses surface tension to walk on top of water. And that's because of the cohesion of water molecules. The cohesion is due to hydrogen bonds. So the hydrogens in the water form bonds with hydrogen molecules in other water molecules. And so they've been binding the molecules of water together. Okay. So surface tension is important. Adhesion is important too if there's cohesion. You think about water on the side of a glass. We'll see this happening in blood, for example, adhering to things like and flowing through blood vessels and capillaries. In addition to cohesion, water also has a really high specific heat. Okay, And this means that the amount of energy required to change 1 gram of water by 1 degree Celsius. Okay. It has a high latent heat of vaporization, which means it actually is a pretty high point before it turns into gas. And compared to other liquids, it expands when it freezes. Okay. And this is why ice floats, right? Because as water freezes into ice, those ice cubes are going to be lighter, less dense than the water liquid, and so they'll float on top. Okay. And so these are all specific specific features of water that affect how our bodies operate because so much of our body tissues are made of water. Water is also a really versatile solvent. And we'll see this too, particularly for things like proteins. Proteins are going to dissolve readily in water. And so when we're carrying proteins in our blood, for example, we'll have proteins dissolving or being solvent in the water in our blood. Okay. So here's an example showing about the hydrogen bonding, right? Here's 1, 2, 3, 4, 5 water molecules, and how that cohesion is created by the hydrogen bonds holding those molecules together. And we said that water is a solvent. So here's water dissolving table salt, NaCl, into sodium and chloride. Okay, so it's dividing it out into individual molecules there. Okay. In biology and in bodies, we have to think about building and breaking down stuff, right? This will become really important. We start thinking about things like digestion and metabolism, is that we're going to be talking constantly about building things up and breaking things down, okay? And so why this is in this water section, is that these are some of the really simple and important reactions that take place in our bodies, okay? A dehydration synthesis reaction is when here you have 2 glucose molecules. Let's say you have 2 glucose molecules, you want to put them together to make maltose. Well, when you do, when a bond is created between the 2 glucose molecules to make maltose, what's lost is H2O. So that means that the other product of this reaction, besides maltose, is a water molecule. Same thing happens here. Glucose plus fructose. When you put those together, our building reactions are called anabolic. You can think like anabolic steroids, right? A building reaction, anabolic reaction of glucose and fructose, means that when you put them together to make sucrose, you get a water molecule. So a dehydration reaction is any one that has water as a product. Okay. The opposite, where you have to put water in to the reaction, so where water is a reactant, it's called a hydrolysis reaction. So we're adding water to break things down. Here's starch, a big old carbohydrate. When you add water to starch, that water breaks the starch down into 2 maltose molecules. If you take maltose and add water, that will break down into 2 glucose molecules. So it's a dehydration reaction when water is a product. It's a hydrolysis reaction when water is a reactant. Okay. So let's talk about acids and bases. We'll talk about this a lot in one of our later chapters. We talk about blood pH. Okay, so this is sort of an intro to acid and base. We'll talk about this a lot more later on in the semester. So what makes something an acid or a base is what it's doing with protons. So protons are hydrogen ion, and acids are compounds that release protons, hydrogen ions, in a solution. So they're proton donors. Okay. Bases are going to lower levels of H plus in a solution because they are accepting protons. So it's all about movement of protons. Are they being donated or accepted? And that determines which kind of molecule you have, an acid or a base. Okay? So some of the common ones we see in labs and potentially in our environments are going to be things like hydrochloric acid, HCl, that's what we have in our bodies, right? Phosphoric acid, nitric acid. Again, why they're all acids is that means that they are releasing protons. Okay? Bases that are lowering the H plus levels of a solution, okay, are going to be things like sodium hydroxide, NaOH, potassium hydroxide, calcium hydroxide. So these are all going to be bases because they are proton acceptors. All right, so pH, super important. What it tells us is that it tells about the concentration of protons in a solution. So our pH scale runs from 0 to 14. And pure water is neutral. So pure neutral is a pH of 7. Maybe you remember from our first lecture that our blood pH is not 7. It's not neutral. It's slightly basic, meaning it has it's slightly above 7, so 7.35 to 7.45. Okay, slightly basic. Acids have a pH of less than 7, so anything a pH from 0 to 7 is an acid, and bases are greater than 7, so 7 to 14 are considered a base. So our blood is slightly basic. All right. So when you add an acid and a base together, it gives you a salt. Okay. For example, you take hydrochloric acid, HCl, add sodium hydroxide, NaOH, and what do you get? You get NaCl and water, table salt and water. Electrolytes, you might have heard about maybe in sports drinks, are things that dissociate and conduct electricity. Okay. What this means then is that some salts can be used as buffers because they're able to donate and dissociate in different components and buffer changes in the pH of a solution. Okay, so it's not a table that you need to memorize, but just to show you what this pH scale looks like. Okay, so here's our pH, 0 to 7, or 0 to 6 are acids, neutral is 7, 8 to 14 are bases. So if something is more acidic, it means it has a lower pH. If it's more basic, it has a higher pH. But it's a log scale, which means that very quickly our proton concentration increases. Okay, so let's talk a little bit about buffers. Buffers are really important because just like in a solution, we need to buffer changes in pH of our blood. Okay, so buffers are molecules that are going to slow changes in pH by either combining with or releasing protons. So we can think back to homeostasis, right? Just like anything else, pH of our blood is going to have a set point. If we want to keep our range small and vary around that set point, we need to have ways to slow the changes in pH. We don't want huge increases in blood pH or huge decreases. We'll talk about why it's dangerous later on in the semester. So what happens then is that you have buffers in your blood and other places in your body that are going to slow the changes in pH by either combining with or releasing protons. Okay, so buffers are composed of a weak acid and a weak base. Like the bicarbonate buffering system in our blood, we're trying to keep our set point and our range of pH in our blood between 7.35 and 4.5. And this is actually one of the systems where we have a really tight control over this because just that 0.1 is our normal range. And if we get below 7.35, that's considered acidosis. Your blood is too acidic, and then you can have some problems. If blood has a higher pH than 7.45, you get alkalosis. Again, dangerous. So we have very tight control over our blood pH because of this bicarbonate buffering system in the blood. Okay. And so here's something you will need to memorize eventually, maybe not yet, but soon, where we see that here's water and carbon dioxide. Okay. And remember that these are some of the waste products of metabolism. Right? So we're getting these things all the time in our bodies. And here's bicarbonate. Okay, that's that buffering system. And here you have protein here and this carbonic acid, proton and carbonic acid. Okay, so we'll talk about this in detail later on in the class as well. Okay, so what happens then is that this reaction can go in either direction depending on the concentration of protons in your blood. So a pretty cool system because if you need more to go one way versus the other, you can. Okay, so that was very, very briefly about pH, acids, and bases. Again, we'll cover that in great detail in later chapters. Okay, so now we're going to get into talking about some of the other macromolecules found in the body. There's 4 major components, 4 major types. So carbohydrates are the first one. Okay, so carbohydrates, things we all love. Cell structure and energy sources. Okay, so lots of our foods are directly carbohydrates, but what makes them all carbohydrates is going to be that they consist of carbon, hydrogen, and oxygen with a formula of CH2O and then an N. So something else out here. But this is going to be our basic formula for a carbohydrate. Okay. Our first kind of carbohydrate is called a monosaccharide, and these are simple sugars. Simple sugars contain 3 to 7 carbon atoms. And when you look at a molecule like this, you might say, oh, it's only one carbon. I only see one C there. Well, every bend in this ring is a carbon. So it's just how they draw these chemical molecules is that unless there's something different there, like you see in this ring has an oxygen present, every bend is a carbon. So we can count carbons by saying, okay, there's one carbon here. 1, 2, 3, 4, 5, 6 is a 6 carbon molecule. Okay, so this makes it a monosaccharide. Monosaccharides are things like ribose, deoxyribose, glucose, fructose, galactose. You probably recognize some of these because ribose and deoxyribose are components of RNA and DNA, and glucose is our primary blood sugar. Okay, so important monosaccharides. Interestingly, we said that glucose is our primary blood sugar, but glucose, galactose, and fructose have the same chemical formula. They're all C6H12O6. So the only difference then is their structure. They're called isomers because they have the same chemical formula, but they have different physical and chemical properties because of the different structure. So let's look at this. Here's glucose. Here's galactose. The only difference is the position of this OH, this hydroxyl group. In glucose, it's on this side. In galactose, it's on this side. That's the only difference. So it's structural, not chemically different. Okay? In fructose, our difference is in where this double bond is. So this double bond here is down further in the chain. It's not the top, it's in the second carbon. Okay? But again, same chemical formula, still C6H12O6, just a different position of the double bond. That makes it fructose. What means, though, is that these three molecules behave differently based on their structural differences. Okay. So when you put two monosaccharides together, they're called disaccharides. So disaccharides, two monosaccharides put together, we can bind them together with one of those dehydration reactions, right, where we have water as a product of our reaction. And so things like lactose and sucrose are disaccharides, double sugars. We can also have polysaccharides, where you have many sugars bonded together. So you take a bunch of monosaccharides, bunch of dehydration reactions to make a big long chain of these monosaccharides all stuck together. And that makes a polysaccharide, so it's multiple, poly. Many sugars bonded together. Things like glycogen, which is how we store glucose in our bodies. Starch, cellulose, and these are things that we can eat, right? But starch and cellulose are really how plants store glucose in their tissues. And dextrin is also an example of polysaccharide you might have heard of. Okay, so glycogen, really important. We store glucose in our liver and fat and muscle as glycogen. And here's what it looks like. It's a bunch of branches of glucose. So here's a glucose molecule, and by adding them together with a bunch of dehydration reactions, you get these chained branch structures of glycogen. Okay, so that was our brief foray into carbs. Now we're going to get into lipids, and then we'll take a break and I'll send you on to your second video. Okay, so lipids is our second category of macromolecule in our body. Lipids are insoluble in water. We said carbs were soluble in water. Lipids are insoluble in water. They are nonpolar, or they're considered hydrophobic, which means scared of water. And you know this because if you put oil in a cup of water, it separates, right? The blobs stay together as oil because fat is hydrophobic, okay? So fats and oils are our lipids, examples of lipids, okay? And just like in carbohydrates, we can have different kinds of lipids, different divisions. So some are simple lipids. We can have monoglycerides, which are one fatty acid attached to one glycerol. We can have triglycerides, which are 3 fatty acids and 1 glycerol, put together through a dehydration reaction. So you're starting to see some patterns, right? When you're building stuff, you use dehydration reactions to build things. Okay. So here's what a triglyceride looks like. So tri-3-glycerides. There's 3 fatty acid tails attached to a glycerol. Okay. You can put different fatty acids here. They can be different fatty acid chains or the same kind of chain. And where this can matter, especially for things like health, we think about saturated and unsaturated fats. Okay. So a saturated fat is one that has no double bond in the fatty acid and is therefore solid at room temperature. Something like butter. Okay. But what do you think? Is this going to be a saturated or an unsaturated fatty acid? It's a double bond. So double bond means that it is an unsaturated fat, something that's liquid at room temperature, like oil. All right. So different examples here of kinds of fatty acids is oleic acid, allylic acid, okay? And the double bond makes it unsaturated, okay? But it also, the position of the bond can matter. So we've heard about a lot of things about like trans fats in the past few years. And this is what that trans fat means. So it means that the double bond makes this kind of shape. So the positioning of this means that you have, this is a trans bond versus a cis bond. Trans bonds means you have one hydrogen on each side, like this, okay? And so there's kind of, the jury's kind of still out on whether trans fats have a huge effect on health. We're learning a lot more about effects of fats and types of fats on human health. And so it's not really clear whether all that worry over trans fats is really accurate in terms of the science. Okay. We can also have complex lipids, things like phospholipids and cholesterol. And phospholipids are really important because they are what make up our cell membranes. So a phospholipid is going to contain a phosphate group, here it is, here, phosphate group, and 2 fatty acid chains. Okay, so 2 chains here. And so it ends up looking something like this, right? They always draw fatty acid, they always draw phospholipids with this little polar head and these non-polar tails. This becomes really important for how they orient into a cell membrane. Okay. So I said the head is polar, the tails are lipids, and so they're non-polar and hydrophobic. And when you put a bunch of these things together, they make this phospholipid double-layered membrane. It's called a phospholipid bilayer membrane. That becomes really important for how we transport things across cells. Okay. Cholesterol is also a complex lipid. Cholesterol is important in itself. It's another cell membrane component. It also is the basis for all of our steroid hormones. So steroid hormones come from cholesterol and have really important jobs in the body, right? So some of our cholesterol-based hormones, our steroid hormones, are going to be things like cortisol, testosterone, estradiol, which is one kind of estrogen. Other ones include progesterone, aldosterone, and these are all really important for controlling really variable functions in the body. And we'll talk about that when we get to the endocrine system. Okay. Another kind of important lipid is going to be prostaglandins. These are fatty acids that have a cyclic hydrocarbon group. Okay, so they have this 5-point carbon here, and they have lots of regulatory functions in the body. So they're going to be a potent regulator of blood vessel diameter, so vasodilation and constriction is affected by prostaglandins, inflammation, clotting. There's lots of research into prostaglandins in terms of immune function, so we know it's really important for some of these roles in the body. Okay, and finally I want to talk about ketone bodies, which is what occur when hydrolysis of triglycerides releases free fatty acids. Okay. So this can be used for energy. We can take free fatty acids and use them for energy, just like we use glucose for energy. You can use free fatty acids. Or they can be converted in the liver into ketone bodies. Ketone bodies, however, are acidic. Okay, so elevated levels of this can cause what's called ketosis. And this is actually pretty normal when you're burning fat. So if you're burning fat, you might have elevated levels of ketosis. But it can eventually form what's called ketoacidosis. So this occurs when there's ketone bodies in the blood, and there's so many of them that the blood pH is lowered and becomes acidic. And probably some of you have thought about or heard about the ketogenic diet, right? And that's the goal of those ketogenic diets, is to burn fat. And so the argument is, yes, okay, well, you're forming ketone bodies. We want our body to form ketone bodies because we're burning fat. That's fine in theory. In practice, what happens is we get to this place of ketoacidosis way too quickly, and it can be very dangerous very quickly. So low blood pH, that can be very bad. It's also really hard on the liver and the kidneys to process some of this stuff. And so what we find is that with ketogenic diets, they can be good for very specific things, right? Like seizure control in kids, short-term weight loss, it's possible. But long-term, this ketogenic diet is actually very dangerous physiologically because of these kinds of issues. Okay, so I'm going to stop there for now. I will finish your next video talking about proteins and DNA.